I'm Dr. Pramod Nagarao Magar, working as a subject matter specialist in entomology at Krishi Vigyan Kendra Yavatmal, Dr. PDKV Akola, and welcome you all for the 16th lecture of International Training cum Certificate course on precision agriculture, farming with new perspective. And I would like to introduce about today's guest speaker, Kritika ma'am, nutrition pathologist at ICRISAT. Kritika ma'am is working with GMS team as a visiting scientist since December 2017. Her primary research project in collaboration with World Wage is on phenotyping elite genotypes of mung bean for drought adaptation. She is also involved and deriving the mung bean crop growth parameters for crop models and farmer participatory studies to extend the relevant research technologies, solutions, and understand the complexities of farming system. Her other interest includes nutrition-sensitive agriculture, potential of land races and underutilized crops and farming system research. She has supervised about 3 intern students. She has 2 publications from her PhD that focused on particularly phenotyping transgenic chickpea for abiotic stress responses. So dear friends, before we get started, it is my humble request to all, you will have to the opportunity to ask your questions or queries for the today's presenter. You may ask by typing your questions into the chat box. You may send your question at any time during the presentation. Our guest speaker will address them at the end of presentation. So over to you, ma'am. Thank you.
Thank you,
Pramod. Thank you.
Yeah, thank you, Pramod.
मैम आप स्टार्ट कर सकते हैं स्वागत आपका लेक्चर में
जी thank you. I'm just sharing the screen. Can you see my screen?
Yes, ma'am.
Yeah, okay.
Yeah.
Thank you. Thank you. I'm Kritika. Thank you, AgriMeet Foundation, for the opportunity to share our research with such a wide platform. So today I will be talking about the sensor technologies that are applicable for nutrition inclusive agriculture. For a brief, so ICRISAT is part of the CGIAR consultative group for international agricultural research. It is headquartered in Hyderabad and it has multiple locations, research centers in Africa. So the main core interest of ICRISAT is to look into farming systems and now more into the agribusiness as well, to look at how we can make profitable farming in dryland systems. So the 3 main focuses that we have, or which is towards our mission, is to overcome poverty and hunger, reducing malnutrition, and preventing environmental degradation. So in the semi-arid tropical regions, there are several crops that are cultivated apart from rice, wheat, and maize. So in ICRISAT, the focus is on chickpea, pigeon pea, groundnut, sorghum, pearl millet, other millets, and now more on oilseeds and other legumes. So we know that in breeding programs, it takes a lot of years to develop a new variety. And in that process, there is a requirement for phenotyping or screening large set of lines. And out of those screened lines, very few of them get released, and of them, very few get adopted by farmers. So similar pipeline is used for multiple traits. It could be for disease resistance, it could be for abiotic stress responses, it can be for nutritionally dense crops as well. So in this process, we see that when we have a lot of lines, say around 10,000 of lines that we have to screen during the different generations of the breeding pipeline, there is a technology gap, which is where we would like to address and see how we can deploy sensors for this particular technology gap. So when we say crop quality, what we mean is both the grain as well as fodder quality. In this picture, you can see the rice grains that are cultivated by farmers and also the fodder. So in a farming system, both grain as well as fodder is of critical importance. Similarly, it is the same for cereals as well as legumes as well. Some of the varieties are developed or bred for being a fodder crop. In that case, we have to look into the protein content and the fiber content, digestibility of the fodder, and how much of dry matter is available, and such traits for the livestock of interest. So while we look at grain quality for our human consumption, we should also look into the fodder quality for the livestock stability. Yeah, so in this particular presentation, I will focus on grain quality and mostly on organic components. But this slide gives an overview of what are the types of grain qualities that can be measured and which are of importance for the market-related traits as well as for farmers. So one is organic components that involves protein, starch, amylose, fat, oil, fatty acid composition, moisture, fiber content. The inorganic components are more on iron, zinc, calcium. It could also be contaminants like arsenic, lead, and which are more of challenges to quantify. And then physical grain properties such as structural abnormalities, contaminants, and then when you have adulteration, and we can also look into viability now. And there is also processing related properties, which is probably the one that mostly decides the price in the market, which is related to grain weight and moisture. So this is an example of a maize grain, where we see that the endosperm layer is where the starch, protein, vitamins, antioxidants are there, whereas the embryo is where the minerals and vitamins get compartmentalized. The pericarp has certain kind of minerals and fibers that get accumulated, and there is alluron layer where antioxidants also get deposited. So maize has something called zein proteins, which is different from the ones that are there in sorghum grain. So this one is a picture of electron microscope transection of it, where we can see that these circular bodies are the protein bodies that get accumulated on the inside the grain. And this vacuole-like thing that you see is the starch granule. So this picture on the left is of a sorghum grain of a wild genotype. And the right side image is of a mutant genotype, where the protein bodies are of different shape. And the starch granule is also of a different shape. So what they noticed with the wild type is that these proteins are hydrophilic in nature. So they don't really get digested well because they're hydrophilic and they stay separate while cooking and all of that. These irregular shaped protein bodies help the sorghum, this particular sorghum type, to be more highly digestible. Similarly, this starch granule, when it is softer, so it also makes the grain type very soft, which results in having a lot of pest infestation after the harvest. So post-harvest damages in this particular wild type is high, and its cooking quality is also low. Whereas in this mutant variety, you can see in this mutant line, you can see the protein bodies are different as well as the starch granule. So this gives the hardiness of the grain, and the protein bodies increase because they are more hydrophobic now. So then they get into more digestibility. So these kind of transitions, these kind of variety development and all of that is a part of the breeding pipeline, which is of researchers' interest. So a grain has several components. It has carbohydrates, proteins, lipids, minerals, fiber, vitamins, and it also has phytochemicals which can be anti-nutritional. So when we have anti-nutritional property, especially in sorghum grains, with this International Year of Millets, there's a lot of promotion for millets to be a part of our diet. And one of the constraints that millets have to be adopted into the mainstream diet is these kind of anti-nutritional factors and the cooking quality. So for these types, there are several grain processing methods that can be used, which will enable us to develop more products. So these kind of products are termed as value chain in the economics division or sector, where you see that for each of these, for a beverage to be developed from, or to be to make a product such as a beverage from sorghum grain, requires a different kind of a pipeline, and that's called a value chain. Similarly, for making breakfast cereals, or for making tea or noodles or pasta. So each of these products require a different type of grain quality. So how do we identify what kind of grain quality is there is one of the questions, and what kind of processing will enable us to make products that can be adopted by consumers is the next question. So briefly, we can see, like in a quick summary. So there is a need for nutritional analysis in the breeding pipeline for the researchers, so that we can identify specific types of varieties and specific. So if you want to make a protein-rich peanut, or if you want iron-rich pearl millet, or if you want something which is more digestible. So those kind of interest is in the breeding pipeline. Whereas in the value chain, which is the market end of it, so what kind of traits will give the farmers an advantage and give them a little bit of enhanced value in terms of both income as well as diversity of options. So for an example, in this value chain, we have something like, so the groundnut Girnar 4 variety that was recently released has high oleic content in the grains. So when we take it, so when farmers in a district called Anantapur in Andhra Pradesh, when they adopt these grains or this variety, when they sell it in the market, they should get an incentive, at least in the basis of price, for the quality of the produce that they bring to the market yard. So for these kind of distinctions, so right now they have something called K6 variety, which has, say, around 40% of oleic acid, whereas the Girnar 4 variety has around 78%. So now we would require a tool that will be able to distinguish between these two varieties, so that at the market yard, the farmer is able to earn an increased pay or an increased price for this Girnar 4 variety. We believe that such kind of incentives will also enable the farmers to make a shift from older varieties to newer varieties, which are also nutritionally diverse. That way we look at both. So here is an example of protein analysis that we do in the laboratory. So when we say that we want to look at varieties that are of high protein, then it has to go through a lot of tedious chemical processing inside the laboratory to identify the nitrogen percentage. So with the instrumentation advancement, so this kind of apparatus has now become more automated. And it's called a nitrogen analyzer in this particular company that they produce. And then we also have certain other companies which also make NIR instruments, who also have these kind of devices. So I can play the video over here. I hope you will be able to see it. So these wet lab, so these analysis or these kind of instrumentation is called wet lab analysis, where the sample is processed with high precision chemicals as well as methodologies that is accepted worldwide. And there is a standard operating procedure for each of these. And the devices are made in such a way that they can be replicated, as well as you have reliable data that you get. Using this data is when we start looking into how we can increase the throughput of this kind of analysis. So in this, maybe they will be able to do 100 samples in a period of 1 month because of the scale at which it operates. Also, the amount of chemicals that is utilized in this particular kind of analysis causes a lot of cost involvement in this. So each sample for protein analysis would cost around 1,200 to 1,500 rupees. When we have 10,000 of samples, then it becomes very difficult for us to have that kind of a throughput. When for 100 samples you would require to spend a month, it also becomes expensive, time consuming, and a lot of chemicals get used, which is something that we would not prefer, because also the breeders have to make quick decisions in the season to advance their genotypes. So in this process, with a lot of instrumentation and sensor-based devices that are coming up, there are two types of devices that we will be looking into today. One is the bench type, and the other is the portable sensor device. Now, the benchtop device is a stationary device. That means it stays in the lab, and we'll have to bring the plants or samples that are harvested in the field, process them. That is, you dry them, you grind them, and then you scan them in the benchtop device. So the sample needs to be brought to the laboratory. Whereas in the portable sensors, you can carry these to the field, and then you can do the measurements on field. Because it's handheld, it's also connected to your smartphones through Bluetooth, and you can do instant predictions on the field itself. So the same company that made the gel dull analyzer also has something like a benchtop analyzer, wherein you can we can see a demo over here, wherein you place, so on a stationary sample, they bring the samples in, you can grind it, or you can also do the whole grain samples, and then the data gets analyzed, that is the spectra gets registered, and then it gives a prediction based on the model. So I will come to these points of spectra and model in a few slides. This is just to give you an overview of what the device looks like and where is the technology that is advancing. So here they are looking at moisture, protein, ash, and I think glutamic or something like that. So this is a benchtop device. Whereas the company that we are working with here looks at portable devices, which takes the chemical testing to the field itself. So here's an example where they're looking at wheat's grain protein quality using the portable device. So the device is called a HoneLab Red, and it's connected to the mobile phone, and it's very handheld, and you can, so it has both sets of, you can use it for floor as well as for grain. This one is when they have brought the sample to the lab, but we can take it to the market yard as well. So in this, they do multiple scans. So they do around 7 to 8 scans, and then you get the result on the device instantly. So here would be the output where they see that you can see the value over here. So it will give the protein content instantly. So all of these two devices that I just now showed are based on near infrared spectroscopy. So this one enables us to look into the organic composition of grain as well as fodder or forage. So the visible range is from 400 to 800 nanometer. And these devices range till 2500 nanometer. So the benchtop devices ranges from 400 to 2500 nanometer, whereas the portable devices start at a much later, this one, around 13,300 to 2,500 nanometers. So in this example, we are looking at, this is across a wavelength. This is from 1100 to 2500, and this is the absorbance. So when the sample is placed inside the sample holder, there is light that is irradiated, and there is the samples will absorb certain amount of light and reflect the remaining. So in this case, they are documenting the absorbance. That is, they detect the reflectance and they show what is the absorbance. So this is how a spectral signature of a grain cereal looks like. And these peaks are related to certain bonds. That is like a CH bond, an OH bond, a C double bond O bond, and those things. So each of these bands also go through a lot of vibration based on the energy that we are exposing them to. So based on these vibrations, these kind of spectral signatures are caught. Where do we come in is when we look at these grain samples, we take the spectral signature of it, we send these samples to wet lab analysis, get their value. When these two data are collected, then we start the chemometrics process, wherein we process the data, the spectral signature, so that we get specific peaks that can be related to specific bonds. And then this combination of bonds is what gives us the quantity of, say, protein content or fat or oil content. And then we build several models. We use either machine learning or statistical methods, and we show the sample. So it is like to say that if this is sample A and sample B, and then we have, say, around hundreds of samples like this, we train each of those to say that if the spectra looks specifically like this, that means the protein content is 10%. If the spectra looks like the blue one, then it is around 2%. And then we train these models so that when we show an unknown spectra, then it should be able to predict. This is very similar to the biochemical analysis that we do in laboratories, where you have a standard curve and then you build it on known concentrations, and then you show the unknown sample, and then you predict the concentration of the sample. The concept is the same here when we look at models. So this is an example of a protein model that we built using multiple crops. So it has several cereals that are sitting here in blue and green, and several of legumes which are in yellow and red. So on the left side is a graph that is built on a training set of, say, around 300 samples. And we take a subset of it, say around 700 samples, and we do it in cell and test if that equation is able to predict the unknown samples. And the throughput of these, when we use it in such devices, is around 150 samples or 200 samples per day, which compared to the wet lab, we would not be able to achieve. So where would these get deployed, such kind of sensors? One is in the research pipeline. So we will focus only on the left side here. So where we look at the quality of crop produce. So when we have a lot of crops coming from the field and high throughput phenotyping platforms, we can use mobile sensors. When we have thousands of samples to be screened, then mobile sensors would be the quick one to do. From there, we can filter out samples that are very contrasting or of interest, and then bring it inside the lab to analyze it in a benchtop analyzer. And from there, which we want to cross-confirm or which are interesting or in the extremes, then we can take them to the wet lab to confirm it. This organoleptic one is mostly to talk about. So even if certain products are rich in protein, they might not taste good. So that is when this comes much later in the value chain, where we want to look at how the variety is going to be adopted by consumers. So this is in the research pipeline that I'm talking about, where the breeders want to make selection for nutritionally dense grains. The other end is sorry, I think you missed me. Shall I start from the same slide?
हाँ जी मैम, स्टार्ट करिए आप।
जी, okay, sorry, I think my internet dropped. When we have these kind of sensors can be deployed in research stations and research institutes. Maybe I'll stop my camera. And the other end is the value chain, where the farmers bring their produce to the fields and then into the market yards, and they get a price for the quality of their produce. So
Slides are not visible.
Sorry?
Please share the slides, it's not visible.
Yeah. Okay. So, yeah. So I was saying in this particular example, so in this slide, so this is for the value chain where these sensors can be deployed. So in market yards where farmers bring their produce, for us to rapidly detect their quality so that we can give them an incentive for the quality of produce or pool similar samples together for the food industry, we would require certain tools which are presently absent. So we propose that such handheld or portable devices will enable us to ensure that the price is paid right for the farmers who bring in, for the quality of produce that they bring, And the food processing units can also have these kind of equipments in place so that the value chain is enhanced based on the nutritional quality. So in summary, we look at several sensor technologies for different components of a grain. So for organic components, we look at NIRS, that is near infrared spectroscopy and Raman-based technologies. For inorganic components, we have XRF. And for physical grain properties, we are now looking into computed tomography, which is also X-ray based. And for processing related traits, it would be gravimetric, which is weight based, and which will also give about moisture. So these three sensors, my colleague Taranya would be speaking about, sharing about them with you tomorrow. Yeah, that's where I would like to conclude. Thank you. We can open for questions now, or I can.
रोहित जी चैट बॉक्स ऑन करिए आप.
Okay, sir.
Thank you, Dr. Kritika. This is Dr. Soni Gupta. We enjoyed hearing you out. Request all the participants to put forward their queries in the chat box so that we will be able to answer those queries. The first question which I am seeing is what is the okay, one sec, ma'am. What is the accuracy of this portable device?
Yeah, so the accuracy depends on the traits that we are measuring. And say, for example, for the protein thing, we are getting around plus or minus 1%. And for oil, we are at around 1 to 2% right now.
Any other question from the participants? The next question is from Harinder Singh. Madam, could you please tell some handheld and lab devices for soil testing?
Yeah, I don't know about lab devices. I think there are some analyzers like that. They do look at nitrogen, phosphorus, and such grains, such elements. But the handheld device, this company Hone does make some. So it's called Hone Black.
One of the comments from J. Jyoti is, can be used for oil palm too?
Yeah, anything that you extract, anything that can be ground and scanned, it can be. Right now, they are also exploring into fruits, which is like a whole fruit that you show the sensor to.
Vishesh Vedoria has asked, what is the price of this device?
Yeah, so the handheld device that we are testing right now is of research grade. So it costed us around 9,000 Australian dollars. And the benchtop is more expensive. I think it's around $40,000 if I'm not wrong.
Okay.
But these are usually one-time investment. And when we have a lot of samples to scan, then the cost gets adjusted as compared to the wet lab, which we have to do too many in them.
Dr. Ritu Sharma has asked, thanks, ma'am, for the wonderful session. One sec, ma'am.
Yeah.
Do the nutritionally fortified cultivars have capacity to compensate for low fertile soils?
Oh, yeah, I'm not the right person to answer this. I have not worked with low fertile soils and their interaction with grain quality. But Dr. Sunita is on call. Maybe she would know better.
Yeah. Dr. Sunita, can you please comment? I guess she's not there.
Yeah.
Anyways,
I'm there. Can you repeat the question? I couldn't hear it.
yeah, one of the participants had asked, do the nutritionally fortified cultivars have capacity to compensate for low fertile soils? I guess the question that the participant wants to ask is whether these nutritionally fortified cultivars be grown on low fertile soils or not.
I think this is not simple to answer, and there is a lot of complications in that. So, nutritionally fortified crop growing in low fertile soil, whether it is below the threshold, then even the best mechanism cannot help the plant to extract extra nutrient.
For growing cells, is it used for all types of crops? I guess the handheld devices.
Yes. Yeah. I mean, all types meaning, yeah. They say that it can be used for fruits as well.
The same question
Yeah.
has been asked by Ishwar Reddy sir, and he has asked, can we use this device for crops like horticulture crops?
Yeah, it can be used. The question is what rate we are looking at, and do we have somebody who can do the wet lab analysis for the particular trait we are looking for?
Yes. Another question asked by Akshay AJ is, are there any Indian companies that produce these types of portable sensors?
Yeah, there's one in Punjab. I forget their name. It's not this small, but it is still portable.
Okay.
Agree something their name was.
Okay.
Yeah.
But Indian startups have started making such kind of sensors.
Yes,
That's good news.
Yeah.
Another question addressed is how this technology will help in horticulture farming.
Yeah, it depends if you are having products that require, say, in case of something like a bricks number or sweetness or those kind of traits. It depends what crop and which trait fetches more money in the market, or which one is of interest to the researcher or to the market.
I would like to add a little bit what Dr. Kritika said, that this technology has potential to trigger the quality-based payment. If quickly you can test the nutritional or any quality of your produce, then you can demand higher cost. Same on the other side, also it can be paid at higher cost. So this e-quality, e-NAM, those all initiative of government can be triggered. Then this technology is not at a scale. It is not well adopted even in research stations. So it coming to a farmer level, it will take some time. But the moment it will come there, it will trigger the quality-based payment.
Yes. Yeah, definitely. More information to the consumers, and so more payment to the farmers. Yeah. So next question by Dr. Trilognath Rai is, zein protein is present in maize. Which amino acids are present in maize? What is essential amino acids? That's a basic question, I guess. But would you like to answer, Dr. Kritika?
Yeah, I actually haven't worked with maize, so I don't know which amino acids are present, I think, and I don't know which is highest. I think all would be, but I don't know which is of specific interest or which one is actually critical there.
He's asked, what is essential amino acids?
Well, within the same protein, I have no idea actually.
Yeah. Okay, you've got a lot of appreciation, madam. Thanks, ma'am, for wonderful session. What is the next question is, what is the life period of this device?
For these sensors?
Yeah.
Well, it depends on the lamp, but then they are replaceable. So the only critical thing is, because the sensors keep getting advanced, like they keep getting improved, the instrumentation part. So that is when you'll have to upgrade the device. Like the benchtop machine, I think it lasts for, I think easily for more than 10 years. But then they will also improve their device. The lamps get better, the small parts get, so.
Yes, true. So people have asked, please share PPT. Ma'am, please
Yeah.
share PPT.
Yeah.
Very nice and useful presentation, ma'am.
Thank you.
And ma'am, could you please mention the current horticulture crop this device is being used?
I'm not sure, but then I think somewhere in they're using it in vineyards. So for grapes, I think they're using it. This particular hone device.
One of the question is, what is the rate of adoption of this technology in our country at farmer level? It's
Oh, yeah.
quite low, but
Yeah.
Yeah.
So these technologies are at present in research institutes which haven't cut across even there. And ideally we would want, so this would not come down to farmer level. I don't think the farmer needs to check the quality of his produce. It's the procuring agencies who will have to check the quality and pay the price for it. So the device wouldn't make much sense for the farmer's level. So
Another
right now,
another question Gopi Krishna ji has asked, does this device need calibration with respect to different samples?
yeah, it depends. Yeah, so right now we are looking at cereals and legumes. So the device, so the device doesn't require calibration, but the models or for the traits we need to make calibrations. Also in the hardware side, we can optimize it for different grains. So some grains are very bold, some grains are very small. So based on that, the sample holders can be optimized. But the calibrations are built for the traits like oil, protein, moisture.
Okay, fine.
Yeah, so
Dr. Ankur has mentioned about the essential amino acids present in maize. I guess lysine and tryptophan he has mentioned. Yes, these are the essential amino acids present in that. Kesav Kumar has commented, short and very informative session, ma'am. Thanks a lot. Ma'am, can these devices detect the seed-borne diseases? It is asked by Vedum Ishwar Reddy.
Diseases, no. I think for that computer tomography or X-ray CT will be useful. Seed-borne disease, I am still not sure whether it will, even CT, how much it will path. Okay. So yeah, I don't think this will not be able to tell that.
Okay. Rishikesh Vadoria has once again asked, can you please explain more about how to analyze the spectral data?
Yeah, maybe I can show the slide and then we go from there. Yeah. So between the data collection and building a model, there are a few steps that we follow. One is to do the data pre-processing. So this one has a lot of statistical methods that are used. So the FOSS company comes with its own proprietary software called WinISI and Calibrator Pro. But basically what we try to do is to apply derivatives to these spectral signatures which are documented, and so that we can enhance the peaks. Ideally, what we want to do is to distinguish these kind of wavy structures so that we will be able to identify that this kind of vibration is coming from an OH bond or a C double bond O bond or a CH bond, NH bond. So we can do a first derivative, second derivative, we can do smoothing, and these kind of several statistical tools can be applied for sharpening these bonds. And from here, you can use a lot of like PCA, you have partial least square PLS, you have modified PLS, and then you also have CNN, ANN, and now with these machine learning, you have gradient boost and all of those kind of random forest methods. All those algorithms can be applied one after the other also in a stacked form so that these signatures become very enhanced and these kind of adjustments are made where each sample is designated to one particular quality or percentage. Say in this example for protein. So then it kind of distributes the sample. So here after doing this data collection, say you have 100 samples. From here, I would take 70 samples, use it for training the sample, that is to make this kind of a model, and then keep 30 samples independently, which is not shown to the statistical algorithms at all, and use it for testing. That is the graph over here. So this is what I use for training, where I have a larger data set, which I'm training the model to say that this particular spectral signature is associated to say 10% of protein, and then use this kind of a model to predict the unknown data set. So this gives me a idea of how good the model is working. So yeah, does this help, Rishikesh?
Thank you, Dr. Kritika.
Yeah.
Thank you for explaining this out.
Yeah.
And I hope there are no further questions or chat. Yeah. Thank you, ma'am. People
Yeah.
are thanking you, ma'am. And yes, ma'am, thank you for that explanation you have done. I'm seeing the replies coming in the chat box.
Yeah.
Chat box
Thank you,
again.
Okay.
Okay.
Yeah.
I guess it's time to wind up now. And people are still mentioning the awesome presentation you did now, ma'am. Thank you for sharing your expertise and years of knowledge on these sensors. It was really beneficial to the people or to the participants here.
Yeah.
Thank you for the wonderful lecture, and we like to keep on collaborating further and increasing the wealth of knowledge.
Yeah.
We'll love to have you on this platform again and again in years to come and months. We'll have wonderful collaboration. Actually, we are looking forward to it.
Yeah, thank you very much for giving us the opportunity, and also for the entire team. So tomorrow when my colleagues are presenting, I think there will be more on sensors and phenotyping.
Definitely. We'll be looking forward to it. And thank you, Dr. Sunita, for joining here. I'm following all your lectures, ma'am. Due to some busy schedule, if I'm not present, please don't think that I'm not following your lectures. It was wonderful to be on board with you. So thanks again, and I'll be signing off now. Thank you, participants. Please be in touch through the WhatsApp group, and we'll be letting you tomorrow's lecture schedule. Thanks, and have a good evening. Bye.
Thank you. Thank you all.
Bye.